Welcome, Brookings Bio students. This is Mrs. Rydell. In our last slideshow video, we looked at how scientists discovered DNA was the genetic code molecule, and we talked a little bit about what the structure of DNA looks like. And in this slideshow, we're going to focus more on once we know the structure, how is that structure going to help the molecule, the DNA molecule, do its job in the cell. And we're going to start out talking about something called replication. If you think back to what we learned about DNA in our mitosis chapter and in our cell parts chapter, we talked about how DNA in a prokaryote is different than DNA in a eukaryote. Remember when we colored our cell parts and we did a plant cell and we did an animal cell and a bacterial cell, and we color-coded all those cell parts. We talked about the DNA that's found in a bacterial cell. Remember, prokaryotes have a single chromosome. It's a circular loop shape inside the cell. And remember, prokaryotes don't have a nucleus. They keep their DNA in the cytoplasm. So it is found out in the cytoplasm of the cell. No nuclear membrane in a prokaryote. If you look at the comparison between prokaryotes and eukaryotes, now in eukaryotes, DNA is packaged into little bundles called chromosomes. And humans have approximately 3 billion base pairs, 60,000 to 100,000 genes. I think that number has been downgraded a little bit. We're talking probably between 30 and 60,000 genes in a human. If you think about taking that chromosome and stretching out the DNA into a line, right? If you look at the DNA inside a chromosome, basically it's one long piece of DNA. If you increase the size of a piece of DNA, so the diameter was as wide as, think, fishing line. I think you know what fishing line is. It would stretch almost 14 miles long. That's a pretty big, long molecule. And think about if I was trying to take that long piece of DNA and pack it into the nucleus of a cell. There's a lot of packaging and scrunching down that has to happen for the DNA to fit into the nucleus of a eukaryotic cell. So think about it. If I wanted to take a string and put it inside of a container, the way you'd have to do that is to coil and scrunch and compact it down into a shape that's going to fit inside. And that's what the cell has to do with the DNA that's in the nucleus of a eukaryotic cell. Remember, in eukaryotes, we have multiple pieces of DNA, multiple chromosomes, and the DNA is found in little linear bundles rather than a circular. And in eukaryotes, we're going to put our DNA in the nucleus of the cell. So if you take a look at the DNA, the structure of a DNA, here's my twisted double helix ladder now. In order to pack it into a chromosome shape, you're going to have to scrunch that DNA really, really, really tight and coil it up. So if you look at the structure of a chromosome, chromosomes are actually made of DNA plus proteins. And there are little proteins inside the nucleus that help the DNA molecule wrap up and scrunch smaller. And those proteins are called histones. If you remember back in our molecule chapter, we talked about all the different things that proteins can do. And we said some proteins are structural, and histone proteins are going to help the DNA molecule compact down into a chromosome shape. The DNA, together with those histone proteins, form these little bead-like bundles called nucleosomes. That's a vocab word. These little bit, and look, it's got those little histone proteins, and the DNA is actually wound around those to make a little bundle. If you've ever seen a garden hose in your yard, and we use the little winder to wind it up on the wrap it around the little hose winder, that's kind of what the histones do. They take the strand of DNA and they wind it around the histones to kind of wrap it up and condense it smaller. Now I'm going to take those little nucleosome bundles and pack them together very tightly, wrap them up, and start making fibers. I'm going to coil up those nucleosomes to make a little bundle here, and then I'm going to coil the coils, supercoiling, and eventually I'm going to pack that DNA down into my chromosome shape. Okay. Remember, when the cell is not dividing, some of the parts of those fibers are unwound a little bit so I can get at the DNA. And remember, the cell needs to be able to read the DNA to know the information about what it's supposed to be doing. So when the cell is not dividing, we see that the coils unwind a little bit, and we call it chromatin. Remember, we talked about chromatin and chromosomes, and the cell can switch back and forth between those two forms depending on whether I need to read the information in the DNA or whether I want to move it when the cell is dividing. If I want to move the DNA around, for example, during mitosis, it's easier to move if it's packed into little tight bundles. And so packing it very tightly, we call it chromosomes. And the cell is going to switch back and forth between those two forms. We talked about those vocab words in our cell parts chapter. We talked about them in our mitosis chapter. So if you look at the structure of DNA now, the structure, remember, structure relates to function. How the shape of this DNA molecule is going to explain how the cell can make a copy of its DNA. If you look at how the molecule is put together, each of the strands, remember, this is a double-stranded molecule, twisted ladder. Each strand has the information it needs to make the matching half. Remember, if you remember our base pair binding rules, Chargaff's rules, A's always go with T's, G's always go with C's. So if you look at the structure of DNA, if I separate the two sides of the ladder, there's information here that can tell me what to fill in to make a matching copy. And that's what the DNA does. We're going to separate my two strands, separate the strands of DNA, and then I'm going to use the bases that are here on the strand to fill in the match. And if I do that on both sides, I use this strand to provide the information to fill in the match over here, and I use this strand to provide the information to fill in the match on this side, I'm going to get two pieces of DNA that are exactly identical. We're making copies. So all I have to do is open the DNA to pull those two strands apart and then use those as what we call a template to make the copy. The site where the strand is going to separate and where I'm going to start copying the DNA is called a replication fork. Replication, replica, is a copy. We're making a copy of the DNA. And notice how I go from a double-stranded ladder to split apart. That's the fork. Think of it like a fork in the road. The replication fork is the place where we're going to pull apart those two strands and start copying the DNA. And we're going to have enzymes that help replication happen. Remember, in our enzyme chapter we talked about enzymes. Enzymes are molecules, usually proteins, that help chemical reactions happen in cells, and this is a chemical reaction. So we're going to use some enzymes. First of all, an enzyme that's going to run down the DNA and unzip by breaking those hydrogen bonds. Remember, hydrogen bonds are holding those two strands together. They're between the nitrogen bases. And so we're going to unwind the DNA and break apart those bonds that hold the two sides of the ladder together. And then we're going to use another enzyme called DNA polymerase to join those little nucleotide subunits together to make a match, make a copy. And we're going to use our strands as templates. Remember, in our enzyme chapter, we talked about enzymes are molecules that help chemical reactions happen, and oftentimes enzyme names end in ASE. We talked about that if you go back and you look at your enzyme slideshow from chapter 1. And remember, we said a lot of time the beginning part of the name tells you what the enzyme is going to do. This enzyme is going to make a polymer out of DNA. Polymers, remember, are made by joining lots of little subunits together to make a bigger molecule. This enzyme is going to join those nucleotide subunits to make the DNA copy. And as it does that, it not only joins the nucleotides, but it does a little spell check. Make sure it's putting the right matches in. A's always go with T's, G's always go with C's. And now that copying is going to happen in opposite directions. Remember, our two strands run opposite directions from each other. And they're going to happen in multiple places at once on the chromosome. If I just start at one end of the DNA and run all the way down that long strand, it's going to take a really long time to copy that one little piece. And remember, we're copying 46 chromosomes in a body cell when we're doing mitosis. So if I want it to happen a little faster, I'm going to open multiple replication forks all along the DNA. And then once we get openings and start making copies, the copies are going to spread down the strand until those replication forks bump into each other. And it goes a lot faster if I'm starting my copies in multiple places. So we're going to watch a little animation about how this process of replication works.
Deoxyribonucleic acid, or DNA, is a molecule made up of two long strands. The strands wrap around each other, forming a double helix. The DNA double helix resembles a twisted ladder. The sides of the ladder are formed by alternating phosphate and sugar groups. The sugar and phosphate groups form the backbone of a strand of DNA. The sugar in the DNA backbone is called deoxyribose. The two backbones in a DNA molecule face in opposite directions. Pairs of bases form the rungs of the DNA ladder. Hydrogen bonds hold the base pairs together. There are 4 different bases in DNA: thymine, adenine, cytosine, and guanine. The names of the bases are often abbreviated. T stands for thymine, A for adenine, C for cytosine, and G for guanine. T always pairs with A. C always pairs with G. A TA pair and a CG pair are called complementary base pairs. Each group of phosphate, sugar, and base is called a nucleotide. DNA makes copies of itself through a process called replication. At the beginning of replication, the double helix unwinds. Enzymes break the hydrogen bonds, holding the base pairs together, and the strands separate. Each strand of DNA is copied by an enzyme called DNA polymerase. Each old strand of DNA serves as a template for a new complementary strand. The new strands are assembled using free-floating nucleotides. Complementary base pairs are matched. A always pairs with T, and G always pairs with C. As each new nucleotide is added to the chain, covalent bonds form between the sugars and phosphates along the new backbone. One new DNA strand is continually assembled base after base from end to end. The other strand is built in the opposite direction in short fragments. One strand is made in fragments because DNA polymerase can work only in one direction. Because the two backbones in a DNA molecule face in opposite directions, the enzyme must work backwards to assemble one strand. After fragments have been built, a covalent bond is formed between them. The process of replication continues. A's are paired up with T's, and C's with G's, until two identical helices have been formed.
We're going to take a break here and do a little acted out activity by playing the role of those nucleotides in the DNA strand to see how this actually works. Stay tuned in the next slideshow to see what happens next.